So hello students and welcome back to another video tutorial by Genome Biotech. In this video tutorial, we would be talking about the process of DNA replication. How does the process of DNA replication take place? In what manner the process of DNA replication occurs? So DNA replication do not starts at random location in the genome. So DNA replication just do not begins at the random location within the genome, but it always takes place at a specific location within the genome, and the location where the process of DNA replication takes place is referred to as origin of DNA replication. So there are very well clear cut marked areas within the genomic DNA where the process of DNA replication starts. Those areas are referred to as origin of DNA replication. Okay, the DNA whose replication starts from an origin and proceed bidirectionally or unidirectionally to the termination site is referred to as a replicon. When you talk about a replicon, what is a replicon? A replicon refers to as a unit of DNA replication. Okay, a part of dsDNA, a part of double stranded DNA, in which the process of DNA replication begins from an origin and proceed bidirectionally or unidirectionally to the termination site is referred to as a replicon. The segment of DNA that is involved in the process of DNA replication is referred to as a replicon. Okay, so prokaryotic replicons are usually circular because prokaryotic DNA which is present is usually circular in nature. So if you look here, this is the prokaryotic DNA molecule. Okay, so the prokaryotic DNA molecule which is present, this prokaryotic DNA molecule is usually circular in nature, whereas the eukaryotic DNA molecule, whereas the eukaryotic DNA molecule is usually linear. This is the double-stranded DNA molecule which is linear, and it is always closed at its end. Every eukaryotic DNA molecule is closed at its ends. Its ends are never left open. This is the type of DNA that is referred to as the linear double-stranded DNA, which is present. Linear dsDNA, which is present in the case of eukaryote, and this DNA is called as cccDNA, cccDNA molecule. Okay, cccDNA molecule. So these are the two different types of the DNA molecules which are present. In bacterial cell, the circular chromosomes contain a unique origin and a DNA replication proceed bidirectionally from the origin to the terminus. Okay. In the case of bacterial cells, now the number of origin of replication in each and every cell may vary. In bacteria, there is a single origin of replication that is referred to as a unique origin of replication, and from this origin of replication, DNA replication is going to proceed bidirectionally. Whereas in the case of eukaryotes, on the other hand, eukaryotes have many number of origin of replication, and these are said to be multi-repliconic in nature. Okay, so the whole bacterial genome is a single replicon. In the case of bacteria, we talk about monorepliconic form of replication. That means the entire bacteria behaves as a single repliconic unit, whereas in the case of eukaryotes, we believes and talks about multi-repliconic unit, in which there are large number of replication sites which are present that will be taking part in the process of DNA replication. Now next point, E. coli, the origin of replication is referred to as Uric. When you look at the E. coli, this is the E. coli origin of replication, and this E. coli origin of replication is commonly referred to as Uric. Uric is referred to as the E. coli origin of replication, which has two different types of conserved sequences. One is referred to as the 13-mer consensus sequences. 13 nucleotide sequences are there, and these are called as 13-mer sequence. E. coli के genome में, in the case of E. coli genome, there are two different types of conserved sequences. One is called as the 13-mer sequence, another one is called as the 9-mer sequence. This is the origin of replication in the case of E. coli. This is called as ORIC in the case of E. coli, also referred to as origin of replication in the case of origin of replication in the case of E. coli, called as ORIC sequence. The ORIC sequence in the case of E. coli is made up of 245 base pair, in which the 13-mer region has 3 repeats and the 9-mer region has 5 repeats. You can look here. These are the 5 repeats which are present: third, fourth, and 5. And the 13-mer region has 3 repeats: first, second, and third. So these are the 3 repeats which are present. So 13-mer region 3 repeats, and 9-mer region 5 repeats are present. Okay, so you can look here. Individual replicon in eukaryotes genome is usually smaller, generally about 40 to 100 kilobase pair in size, whereas the individual replicon in the case of a prokaryote such as E. coli is much more larger. It is much more longer. It is approximately 245 base pair of DNA long. Okay, this 245 base pair of DNA, which is present in the case of E. coli, consists of 9-mer and 13-mer region. Now, why these 9-mer and 13-mer regions are present, and what is the significance about this 9-mer and 13-mer region? See, the binding of proteins is going to take place at the 13-mer region, and 9-mer region are going to be the 9-mer regions are going to be the site for the binding of the DNA A protein. See this. And then after the binding of the proteins which are going to take place in the 9-mer region, the 13-mer region is going to be melted. See, this is what is going to happen. If you look here within the structure of this DNA, then ATP along with the DNA A is going to bind. ATP and DNA A is going to bind. It will form a helical structure. It will form a spiral structure, something like this, like this. Protein will bind within this particular structure, and once the protein will be bounded within this particular structure, then it will cause the melting of the dsDNA to take place. Now, how and in what form the melting is going to take place, please do try to understand. If you have to melt the DNA at a particular sequence such as this, and you have to use this particular sequence, why you are using this particular sequence and you are melting the DNA at this particular sequence? To understand this process, you must calculate the AT and GC content of the 9-mer and the 13-mer region. Calculate the AT content and calculate the GC content. Calculate AT and GC content. Okay, now calculating AT content and calculating GC content. Okay, when you calculate AT content, this is AT 1 2 3 4 5 6. Calculate GC content. GNC would be 1 2 3 1 2 3 4 5 6 7 8 9. Out of 9, you find that there are 3 GC. Okay, so 3 GC out of 9, that means one third GC content means equivalent to 33% GC content. And AT content in this 9-mer region will be approximately 66%. AT content will be 66%, GC content is 33%. Now you look here, what is the percentage of GC content? There is single G and there is a single C out of 1 2 3 4 5 6 7 8 9 10 11 12 13. So the GC content that you are going to calculate here, this GC content will be equivalent to 2 upon 13. A very low value will be there of 2 divided by 13. So the AT content is going to be very high. So this is the difference that is going to be produced. Okay, just calculating the same. Here you get a GC content of 15%. Okay, you get an AT content. That is why you get an AT content that would be equivalent to 85%. Now, what do you mean by this? This means that the bonds in the 9-mer region are much more stronger. This region is much more stronger. Okay. And this region is much more weaker. As this region is weaker, so you use this region as an axle in order to melt this particular region. आप इस रीजन को एक्सेस की तरह यूज करो, क्योंकि इस रीजन में जीसी कंटेंट ज्यादा है, और मेल्ट करा दो इस रीजन को. You use 9-mer region as an axle in order to melt the DNA at the 13-mer region, because the AT content of the DNA at 13-mer region is very high. Opposite is not true, because AT has just two number of double bonds, whereas GC has three number of double bonds. So the bonding in the case of 9-mer region is stronger. You use stronger bonding in order to make a cleft and melt the DNA where it is loosely bounded. Thus, strong region, strong region of the DNA is being used, of the DS DNA is being used, is being used in order to make a swivel, in order to make a swivel and melt the DNA, and melt the DNA at, and melt the dsDNA at the 13-mer region. 13-mer region. Here you would be melting the DNA molecule at the 13-mer region. This is how the melting of the DNA is being performed. In this manner, the melting of the DNA can take place. Any doubt in this particular process? So this would be very clear in your mind why the DNA is being melted at the 13-mer region only. And why 9-mer region the proteins are binding. The first protein that binds at the 9-mer region is the DNA helicase, called as DNase A protein. This DNA A protein causes the melting of the DNA. Here the DNA melting is going to take place. DNA is going to melt, DNA melting is going to take place. Here the DNA melts and the DNA gets converted into an open complex. Okay. Now, the ORI-C contains 11 5' to 3' repeat sequences called as GATC repeats that are methylated on adenine on both the strands. On fully methylation, origins can initiate replication. Hemimethylated daughter strands, which have just replicated, cannot be used again and again for the replication to take place until and unless the methylation state is being restored. See, whenever the DNA is going to undergo replication, whenever a double stranded DNA is going to undergo replication, for example, if this is a double stranded DNA molecule that is undergoing replication, this is the parental DNA molecule, this is the new dsDNA molecule that is going to be biosynthesized. ये वो नया DNA molecule है जो बनेगा. This is the parental DNA molecule that is going to be biosynthesized. When this is the parental DNA molecule that is going to be biosynthesized, then what is going to happen? The new DNA molecule can be biosynthesized over the pre-existing DNA molecule by complementary base pairing. Okay, but the new DNA molecule that has been biosynthesized will be hemimethylated, whereas the old DNA molecule will be fully methylated. The old DNA molecule will be fully methylated, and the new DNA molecule will be partially methylated only. Okay, so this is old DNA, fully methylated. Old DNA molecule is fully methylated, fully methylated. This is new DNA molecule that would be partially methylated. Partially methylated. As it would be partially methylated, so this DNA molecule will be the subject of the action of DNA polymerase enzyme. So DNA polymerase enzyme will perform proofreading only on the partially methylated DNA molecule. Okay, so this is very important. The pattern of methylation is used to estimate that where the proofreading activity is going to take place. Only the partially methylated DNA molecule is subjected to proofreading and never the fully methylated DNA molecule. You must remember that only partially methylated DNA molecule undergoes proofreading. Please look at the chat box. I have provided a comment to you all. See, note. I'm just telling you a note. In case of DNA proof reading activity, DNA polymerase second performs performs proof reading via the screening of nucleotides over the partially methylated strand of DNA, methylated strand of the dsDNA molecule. Considering the fully methylated form as always correct. Now see this. Whenever proofreading activity has to take place. Now, supposingly, if at the place, if in the case of DNA polymerase, second is going to find a mistake. If at the in front of A, there is a guanine residue that has been added here. Okay, now which has to be removed? Either you will have to remove the adenine residue and add a cytosine at its place, okay, or replace guanine and add a thymine at its place. Then which activity will be performed? क्या करेगा हमारा DNA polymerase? What would be the function of DNA polymerase? How DNA polymerase is going to act? Shivani, Hiba, Ankit, try to answer the question. On which strand DNA polymerase is going to change the nucleotide? It will change adenine or it will change guanine. Which nucleotide the DNA polymerase is going to change? Can you answer? Try to give the answer. Hmm? To which nucleotide the DNA polymerase will change? Hiba, Shivani, Ankit. The DNA polymerase is going to work over the lagging strand. It will replace this guanosine nucleotide, and the guanosine will be replaced with thymine. The guanosine will be replaced with thymine. Will be replaced with thymine. Why? With thymine. Because the fully methylated strand nucleotides are always considered to be correct, and the only nucleotides that have been newly biosynthesized, their proofreading takes place, never of the parental strand. प्रूफ रीडिंग कभी भी पेरेंटल स्ट्रैंड की नहीं होती, प्रूफ रीडिंग तो नए बने हुए स्ट्रैंड की होती है. प्रूफ रीडिंग ऑफ द न्यूली बायोसिंथेसाइज्ड स्ट्रैंड टेक्स प्लेस. प्रूफ रीडिंग ऑफ द पेरेंटल स्ट्रैंड do not occur. Is this clear to you or not? Hello. Yes. Okay. Sure. Hiba and Ankit, are you there in the class? Okay. Thank you, Hiba. Okay. Origin of replication in yeast is known as autonomously replicating sequences. So this is all about the origin of replication that is going to take place in the case of E. coli. Then you have the origin of replication that was functional in the case of eukaryotes. it was that of the prokaryotic cells, the E. coli, that how it was functional. Then I have told you what is partially methylated, what is fully methylated, what is the difference between partial methylated DNA molecule, fully methylated DNA molecule, and how does proofreading is being helped by this partial methylation. Now, this partial methylation also ensures, see this, that daughter DNA cannot be used again for the process of replication until and unless they have been fully methylated again. Until and unless they are being fully methylated again, they cannot be used in the case of DNA replication. Now you know about that how the process of DNA replication takes place. Now the next important point, the origin of replication which are being used in the case of yeast is referred to as autonomously replicating sequences. The origin of replication which are being used in the case of yeast are called as autonomously replicating sequences. They are also referred to as ARS sequences. A typical autonomously replicating sequences in the case of E. coli are usually less than 200 base pair in length. They are usually less than 200 base pair in length. That means they're quite smaller. The autonomously replicating sequences consist of an 80 rich regions that include a consensus sequence called as a domain. The other domains that are B1 to B3 domain. A domain contain 14 base pair consensus sequence, contain 11 base pair consensus sequence consisting of 80 base pair. In the case of metazoans, no clearly conserved sequences have been identified. There are metazoans. Metazoans refer to, see, the mammals have been classified into three different types. One is referred to as the prototherians, metatherians, and another one is referred to as the eutherians. In metazoans, no clearly conserved sequence elements have been identified at the replication origins. There was no clearly conserved sequences that were being identified at the origin of replication in the case of metazoans. Some common features of the replication origin are as follows. So, what are the common features associated with the origin of replication in the case of eukaryotes? First, replication origins are always unique DNA sequences that contains multiple short repeated DNA sequences. Okay, these are unique DNA sequences that contain short repeated sequence. For example, the unique DNA sequence is that of 245 base pair, which is present in the case of E. coli. This is present in the case of E. coli. But this DNA sequence also contain certain conserved residues called as 9-mer residues and the 13-mer residues. Okay, so these are the conserved residues which are present. And so you can find the conserved residues to be present here. These short units are recognized by multimeric origin binding protein. These short units are being recognized by specific proteins, and those proteins are referred to as multimeric origin binding proteins. There are specific proteins, and those proteins have the ability to bind to the origin of DNA replication. These proteins plays a key important role in assembling DNA polymerases, other replicating enzymes at the site where replication is going to begin. So all these proteins functions in order to assemble DNA polymerase and other replication enzymes at the site of DNA replications. They are going to work in order to assemble the different proteins at the site of DNA replication. Origin regions usually contains a 80 rich sequence of DNA. Origin regions always contain an 80 rich sequence of DNA. The property helps in unwinding the DNA double helix. Okay. Because whenever there is an AT rich region, this AT rich region can be effectively melted as compared to the GC rich base pair. AT rich base pair can be effectively melted, it can be easily melted as compared to the GC base pair in the DNA molecule. Any doubt, any problem? Any doubt, any problem? Replication DNA, origin of replication must always contain unique DNA sequences. Origin of replication must have multimeric origin binding protein present within itself. Okay, those proteins include DNA polymerases, several different enzymes. Origin of replication must have an AT rich sequence present within itself. Along with the AT rich sequence, the GC rich sequences are also present, which are being used as a civil in order to melt the DNA at the AT rich region. We always know that AT rich region DNA can be effectively melted, easily melted as compared to the GC rich region. Now about the replication fork. Now, what is this replication fork? Is this clear? Any doubt? If you have any doubt, you just ask. If you feel any doubt, you just ask. Hello, any doubt, you just ask. No. Okay. Now, replication fork. Now, what do you mean by replication fork? Now, when you look at the replication fork, replication fork refers to replication fork refers to the area where the DNA replication is going to take place. So if you look at this particular area, this is the area where the process of DNA replication is going to take place. Okay, and this is the direction in which the replication fork is going to move. So this replication fork is going to move in this particular direction. So what is this replication fork? The movement of the replication point constitute the replication fork. The movement of the replication point over the dsDNA constitute the replication fork. Okay, so this is the movement of this particular point in the direction of DNA replication constitute the replication fork. In case of unidirectional replication fork, there is a single direction in which the replication is going to take place. In the case of bidirectional replication fork, there is DNA replication taking place in both the direction. That is in the forward direction also as well as in the backward direction. In case of unidirectional replication fork, single replication fork is being produced. Whereas in the case of bidirectional replication fork, two replication forks are being produced. There would be two replication forks that would be produced. Okay. So here would be the two replication fork, one in this direction and another in the opposite direction. Okay. Now, when you look at a single replication fork, this is referred to as unidirectional replication fork. A replication fork which is being produced only in a particular direction in from the cell is referred to as unidirectional replication fork. When a replication fork is being produced only in a particular direction in a particular manner, then it is referred to as a unidirectional replication fork. When it is going to be produced in both the direction, that means the DNA replication fork is being produced in this direction as well as in the opposite direction, in the forward direction as well as in the opposite direction, then such type of replication fork which is being produced is referred to as bidirectional replication fork. Okay, So this is referred to as bidirectional replication fork. Now, from the origin of replication, you can find there is a continuous biosynthesis over one strand of the DNA. Then this strand is said to be the leading strand, and on the opposite strand there is a discontinuous biosynthesis that is going to take place, and this discontinuous biosynthesis is referred to as the lagging strand. Okay, one is referred to as the leading strand, another one is referred to as the lagging strand. The synthesis in the case of leading strand is continuous, the synthesis in the case of lagging strand is discontinuous. This is how the strands are being biosynthesized. Okay, in the opposite direction, when you are going to look in this particular direction, when you are going to look here, you can find the opposite strand becomes leading, whereas the opposite strand becomes lagging. So just on the basis of the visualization frame, the leading and the lagging strands are being altered. In one visualization frame you find one strand to be leading, एक विजुअलाइजेशन फ्रेम में आपको जो स्ट्रैंड लीडिंग दिख रहा है, ऑन द अपोजिट विजुअलाइजेशन फ्रेम यू फाउंड that the same strand was lagging, and the lagging strand was found to be leading on the opposite side of the replication fork. So we can clearly say that a leading strand is also a lagging strand behind the origin of replication, and a lagging strand just after the origin of replication could be visualized as a leading strand. So scientist says that the leading and the lagging terms are misnomer. In the case of bidirectional replication fork, the one which is leading is also lagging, and the one which is lagging is also leading. नाउ ट्राई टू अंडरस्टैंड इन हिंदी. देखिए जो लीडिंग है बेटा, रेप्लिकेशन फॉर्क के दूसरी तरफ देखोगे ना तो वही लैगिंग है, और जो लैगिंग है रेप्लिकेशन के फॉर्क के दूसरी तरफ देखोगे ना तो वही लीडिंग है. तो लीडिंग भी वही है जो लैगिंग है, और लैगिंग भी वही है जो लीडिंग है. इसीलिए साइंटिस्ट ये बोलते हैं कि टर्म लीडिंग और लैगिंग मिसनोवर है, गलत टर्म्स हैं जिन्हें हम नहीं यूज कर सकते बाई डायरेक्शनल मोड ऑफ डीएनए रेप्लिकेशन को अंडरस्टैंड करने के लिए. वी कैनॉट यूज द टर्म लीडिंग एंड लैगिंग फॉर द बाई डायरेक्शनल मोड ऑफ डीएनए रेप्लिकेशन. इज दिस क्लियर टू यू और नॉट? इज दिस क्लियर टू यू और नॉट? हेलो. ओके सर, ये फायर्स बता फिर से बता दीजिए. ओके. Now this is what I am speaking now. Hiba, firstly I'm going to speak in English, then I'm again going to repeat this out in Hindi, and for all other viewers they can just visualize it again over the YouTube and other visualization platforms. So okay, after understanding the bidirectional replication fork and the process that over looking at the single origin of replication, we can find that on the same from the single origin of replication in one direction there is a leading strand, and just in the opposite direction to the origin of replication the same strand will be referred to as the lagging strand. Okay, the velocity of the fork movement refers to as the speed of the fork movement, and the speed of the fork movement is approximately 1000 base pair per second per fork. The Okazaki fragments is synthesized one in every 2 seconds, and the rate of fork movement is quite slow, just 100 base pair per second in the case of eukaryotes. But in the case of eukaryotes, the process of DNA replication is made faster by the increased number of origin of replications which are present in eukaryotes as compared to prokaryotes. So eukaryotes have larger number of origin of replication. Now DNA replication in E. coli. How does the process of DNA replication takes place in E. coli? The genome of the E. coli is replicated bidirectionally from a single origin of replication. You know that when you talk about the genome of the E. coli, it is being replicated bidirectionally from a single origin of replication. For example, ORIC, E. coli replicon being circular, it do not have any free end. And thus, E. coli always follows a mode of DNA replication that is referred to as theta mode of replication. There are always three different steps involved in the process of replication: initiation, all the steps which are being responsible for the melting of the double stranded DNA molecule and the binding of first nucleotide monophosphate. In order to initiate the process of DNA replication is referred to as initiation. Elongation refers to as all the events that are being involved in elongating the DNA molecules attached at the replication fork. What happens in the case of termination? During the process of termination, at after the process of DNA replication has been completed, after the process of DNA replication has been completed, the the process of termination takes place so that the two daughter strands produced could be effectively separated, one apart from each other. They could be effectively separated away from each other. Then the process is referred to as termination. Now, in the case of biochemistry of DNA replication, we must understand the different enzymes which are involved. So, firstly, we have classified the enzymes on the basis of the phases. Enzymes or proteins which are involved in initiation: DNA A, DNA gyrase, DNA A, helicase, DNA gyrase, topoisomerase second. It is the topoisomerase second which helps in removing the supercoiling. Single stranded DNA binding protein. It will cause the single stranded DNA separated to remain in single stranded form. DNA B is also a helicase. DNA C facilitates the unwinding. Proteins involved in elongation include primases, single stranded DNA binding proteins, as well as DNA ligases. Okay, so those which are involved in elongation are DNA G primase, DNA polymerase, and DNA gyrases. A swivel or a protein to remove the supercoiling, that is referred to as topoisomerases. And the last proteins are the TUS proteins and the DNA polymerase 4. DNA polymerase 4 proteins and the TUS proteins. So these are the proteins that are being involved in the process. Okay. Now, after this, we are going to start about the melting of DNA molecule, that how does the melting of DNA molecule takes place with the help of the enzyme that is referred to as DNA helicase. Now, for this, we would be looking at the animation, and I have already shown you the animation. If you can go with the recorded version of the lecture, I'm going to provide you the recorded version of the lecture. And after looking at the animation only, we will start it from tomorrow class about DNA helicase and primase. Please go through the animation. The link will be shared in your group. From there, we are going to start about helicases and primases and then complete the entire procedure of DNA replication tomorrow only. So that's all for today. Hope you enjoyed the lecture. That's all. We'll get the recorded version of the entire procedure in an animated form through the book Watson in your group. That's all for today. Good night. Bye bye.